So if you printed out your entire DNA sequence, it would fill like 1,000,000 pages with just the letters A, C, T, and G.
Yeah, a literal mountain of paper.
Right. It is this unimaginably massive, infinite bowl of alphabet soup. But here is the crazy part. Just having those letters tells you almost nothing about how to actually build a human being.
Exactly, because you're missing the context.
Yeah, to read the genome, you really need to find the punctuation marks. You need to know which specific letters act as the hidden switches turning genes on, or the boundary walls stopping signals from crossing over.
The master blueprints for the cell, basically.
Exactly. So today, we're taking a deep dive into a groundbreaking new paper about a deep learning framework called BioSeq2Seq. And this AI is doing something that historically was thought to be practically impossible to scale.
It really is a massive shift.
It is instantly annotating the genome. It takes that 1,000,000-page book of raw letters and computationally predicts all the punctuation. It's saving millions of dollars and potentially decades of lab work.
Okay, let's unpack this, because the scale of this problem is kind of wild, right?
Oh, the scale of the bottleneck is hard to overstate. I mean, for decades, finding those punctuation marks, what we call functional elements, meant doing painstaking molecular assays in a physical laboratory.
One at a time.
Literally one single biological function at a time. Because the genome isn't just a flat string of code. It's a dynamic, three-dimensional operating system.
Right. It's alive.
Yeah, you have histone modifications, which act like physical spools that dynamically wind up or loosen the DNA to control access. You have promoters that sit right at the start of a gene to kick off transcription, and enhancers that amplify that signal from tens of thousands of base pairs away. Plus transcription factors.
Which are the proteins, right?
Exactly, the specialized proteins that physically dock onto the DNA to trigger the whole process. So before AI stepped in, how were scientists actually finding these elements? I mean, was someone just sitting in a lab running physical chemical tests on every single gene?
Well, essentially, yes.
Yeah.
They relied on a highly complex physical process called ChIP-seq.
Okay.
You literally have to take millions of living cells, chemically freeze them so the proteins are stuck to the DNA, smash the cells open, and then use specialized antibodies to fish out the specific protein you're looking for.
Wow, that sounds exhausting.
It is. It's highly accurate. But think about the logistics. You need a different, highly specialized antibody for every single thing you want to map, and extensive instrumentation.
Right.
If you want to map all these functional elements across tens of thousands of different species and across all the different tissues in the body, like the brain, liver, heart, it is financially and physically impossible to scale.
I see why computational biologists started looking for a shortcut. The instinct was to just, feed the static DNA sequence into an early AI model and ask it to predict where the switches are.
Right, which makes sense on paper.
But using just the static DNA to predict what a living, breathing cell is actually doing, it feels like looking at a printed roadmap and trying to predict where the live traffic jams are.
That is a perfect analogy.
You know where the physical streets are, but you have no idea if Main Street is closed for construction today.
The static roadmap alone simply doesn't contain enough information. I mean, the DNA sequence in a neuron in your brain is completely identical to the DNA sequence in a skin cell on your arm.
Right, it's the exact same code.
Exactly. What makes them function completely differently is that live traffic, the unique real-time transcriptional activity happening in that specific tissue.
So the older AI just couldn't see the traffic.
When older computational models relied solely on that static DNA sequence, they failed entirely to predict functional markers in new cell lines because they were blind to the live context.
And even when models eventually tried to incorporate live data, the processing power required just brought everything to a crawl. The paper discusses an older standard called DHIT, which used something called RO-seq.
Yes, RO-seq.
And RO-seq is essentially the live traffic camera, right?
Spot on. RO-seq, or run-on sequencing, captures the exact position and distribution of RNA polymerase.
And that's the thing actually reading the code.
Right. RNA polymerase is the actual molecular motor that travels down the DNA strand reading the code. So yes, RO-seq provides a direct, live measure of where transcription is actively happening.
But the older AI struggled with this?
Immensely. Earlier models like DHIT, which relied on older machine learning architectures, like support vector regression, they just couldn't process the sheer volume of that data efficiently.
How slow are we talking?
Well, to predict histone modifications on just one single chromosome out of our 23 pairs, DHIT took anywhere from 3 to 5 hours.
Wow.
And it could only focus on one specific biological mark at a time.
Okay, so for everyone listening, I want to do a quick recap here, just to make sure we're tracking the problem.
Is that right?
Bullet point 1, finding functional elements in DNA physically is just way too expensive and slow.
Yeah.
And bullet point 2, the early AI models either lacked the live context, like they only used static DNA, or they were painfully slow and narrow, like that DHIT model.
That was exactly the ceiling the field hit. And it is the precise ceiling that BioC26 breaks right through.
And how does it do that?
By completely redesigning the underlying engine. It handles this by combining multiple streams of data into a highly advanced tri-modal architecture.
Okay, let's look under the hood of that engine. What are the 3 specific streams of data the AI is synthesizing?
It looks at 3 distinct layers simultaneously. First, the evolutionarily conserved DNA sequence.
The static roadmap.
Exactly. Second, the cell line-specific transcriptional activity from ROSEq. That's the live volume of the traffic. And third, the directionality of that ROSEq data.
Wait, directionality, like which way the traffic is flowing?
Yes. RNA polymerase doesn't just sit there, it moves. Knowing whether the transcription is happening on the sense strand or the antisense strand gives the model crucial spatial orientation.
Got it. I actually have a mnemonic for you, the listener, to remember these inputs. Think of it as RDR for ROSEq, the live traffic, plus D for DNA sequence, the static map, RD.
I like that, RD.
And the researchers actually proved why you absolutely need all of these layers combined, right? Like, they ran tests giving the AI just the live ROSEq data and then just the static DNA data.
Yeah, and the RD combined model crushed them both. The synergy is what drives the accuracy.
Why is that?
Because if you only give the AI the live ROSEq data, it becomes incredibly good at finding active regions of the genome, places where the genes are turned on.
Makes sense.
But the genome also have highly repressed regions where the DNA is packed so tightly that no RNA polymerase can enter. In those silent zones, the live traffic is 0.
So the AI would just see a blank space.
Right. It needs the static DNA sequence to recognize the structural patterns of those silent regions. It relies on the synthesis of both to form a complete picture.
So the data is flowing in, but how does the AI actually digest it without choking like the older models? The paper points heavily to a transformer architecture.
Yes, the transformer is key.
Most of us are familiar with transformers powering text models like ChatGPT, predicting the next word in a sentence. How does that translate to predicting the operating system of a genome?
Well, BioC26 utilizes 11 transformer blocks specifically tailored for genomic architecture. And the true power of a transformer lies in its attention mechanism.
Meaning what, exactly?
Its ability to weigh the importance of distant elements. In genetics, biological functions don't just happen in isolated linear sequences. As we discussed earlier, an enhancer might be located 50,000 base pairs away from the promoter it regulates.
That is a massive distance.
In a straight line, yes. But in the three-dimensional space of the cell nucleus, the DNA strand loops around so that distant enhancer physically touches the promoter.
Okay, so a model with narrow vision would just completely miss that long-range relationship. It needs to see a massive stretch of the genome all at once to catch those distant loops.
Precisely. BioC26 has an unprecedented receptive field. It takes in a massive contextual window of 197,000 base pairs of DNA.
Wow, almost 200,000 at once.
Yes. By analyzing that vast surrounding landscape, it can incredibly accurately predict the regulatory patterns of the central 114,000 base pair region, and it outputs the data at a hyper-detailed resolution of 128 base pair cent.
That is wild. It's like trying to predict the highly specific weather in your exact neighborhood. That's the 114,000 base pair center. But to do it accurately, the AI can't just look up at the...
And giving the AI that wide-angle, state-level satellite view is why it so vastly outperforms the older architectures that were essentially just staring out the living room window.
Okay, time for another quick recap for you listening. Bullet 1, BioC26 uses a tri-modal input, the RD mnemonic, DNA sequence plus ROSEq activity and direction.
A powerful combo.
And bullet 2, it uses a transformer architecture with a massive 197,000 base pair wide-angle lens to get the necessary context.
Exactly right.
But I have to ask, normally feeding an AI that much context slows it down to an absolute crawl. If the old model took 5 hours for one chromosome, how fast is this new engine actually running when put to the test?
The speed is staggering. The researchers tested BioSeq2sec on predicting 10 different types of histone modifications simultaneously.
Oh.
It didn't just show a 14.27% accuracy improvement over the older DHIT model. It processed the entirety of the human genome, all 23 chromosome pairs, in just 2.5 hours.
Wait, 2.5 hours for the whole genome?
The whole genome.
That is a completely different paradigm. But, looking at the paper's data on those histone predictions, there is a fascinating quirk.
Oh, the active versus repressive marks.
Yeah. The model is astonishingly accurate at predicting active marks, like a modification called H3K4ME3. But its accuracy drops slightly when predicting repressive marks, the areas where the DNA is shut down. Why is it struggling there?
You've hit on a really fundamental physical difference in the biology there. Active histone marks are associated with intense, highly localized transcriptional activity.
The live traffic.
Right. In the data, they form very sharp, defined, localized peaks. It's like the AI is scanning a horizon and spotting a distinct skyscraper. It's very easy to define the boundaries.
And the repressive ones.
Repressive marks operate differently. They spread out to pack large sections of DNA tightly together, shutting down entire regional networks.
Like a blackout zone.
Yes. They don't form sharp peaks. They form dense, continuous domains without clear, sharp boundaries.
So instead of looking for a skyscraper, the AI is trying to find the exact edge of a sprawling, flat plateau.
Exactly. And because the live ROC traffic is virtually zero in these repressed plateaus, the AI has to rely almost entirely on the underlying static DNA sequence to guess where the plateau slowly tapers off into a valley.
It's just a much harder mathematical pattern to define.
Significantly harder, but it still does a remarkable job.
So it's incredibly fast at finding these histone switches. But genomes also have structural architecture. What about things like promoters or the insulators that act as boundary walls to stop signals from crossing wires? Can this AI actually map the structural framework of the gene?
It maps them with unprecedented precision. The researchers tasked it with identifying functional elements like promoters, insulators, polyacites, and gene bodies.
And how did it do?
It improved upon previous state-of-the-art methods like DREG by 2.50%, but more importantly, it did it by completely changing how the AI answers the question.
What do you mean?
Well, older models usually treated this as a binary classification problem. They would look at a sequence and guess, is this a promoter? Yes or no?
Okay.
BioSeq2sec treats it as a regression problem. It predicts a continuous signal, essentially drawing a dynamic curve over the genome.
But how does drawing a curve actually pinpoint the exact location of a promoter?
It applies a highly rigorous statistical filter. It's a multivariate, normal distribution peak calling algorithm.
That is quite the mouthful.
It is. But basically, once the AI draws the curve, it doesn't just guess where the highest point is. It checks 5 different dimensions of that peak: the maximum height, the specific slopes on the left and right, and the midpoints.
Okay.
It then runs a mathematical test requiring a p-value of 1e3.
Let me translate that stat for our listeners real quick. That means the AI only officially flags a functional element if the math proves there is less than a 1 in 1,000 chance that the signal is just random background noise.
Yes. It's an incredibly strict filter.
Okay, so that handles the noise.
It removes the biological noise brilliantly. And once it knows where the boundaries of the genes are, the model can actually predict how loudly those genes are expressing themselves.
Gene expression, task number 3.
Yes. In its third major task, predicting gene expression, it evaluated RNA-seq data across 10 different subcellular types. It achieved an AUC, an area under the curve, of 0.9366.
Which is essentially like scoring a 93% on a test, measuring how perfectly the model can separate the true biological signals from the false ones.
Beyond the sheer score, the granularity is what impressed the researchers. The AI was able to clearly mathematically separate the exonic regions from the rest of the gene body.
Exonic regions being the actual coding sequences that eventually make proteins.
Exactly. It learned the internal granular architecture of the gene purely from the data.
Okay, so it maps the architecture, it finds the switches, it reads the expression levels. What about the proteins that actually press the buttons, the transcription factors?
Here's where it gets really interesting. This is where the model pushes the absolute limits of computational biology.
I am ready.
BioSeq2sec predicts 90 different transcription factor binding sites simultaneously, all using one single unified model.
90? To put that in perspective, previously, if a researcher wanted to find the binding sites for 90 different transcription factors, they had to build, train, and run 90 completely independent AI models.
Yes, a massive undertaking.
And this does it all at once. But how does it keep 90 different targets from getting crossed wires inside the neural network?
That was a major engineering hurdle. The researchers found that trying to predict all 90 factors through one single neural pathway caused what's known as gradient interference.
Cognitive overload for the AI.
Exactly. The parameters for predicting one type of protein were actively confusing the parameters for another, so they analyzed how quickly the AI learned each of the 90 tasks and divided them into 3 distinct groups based on their convergence speed.
Oh, I see.
Then they built a multi-branch architecture.
It's like a company realizing that sending 90 completely different projects through one single manager is causing a massive traffic jam. So they split the work. They give the fast, easy projects to one specialized department and the slow, complex projects to another, so the slow ones stop holding up the fast ones.
That multi-branching is what allowed them to maintain performance that rivals the leading single-task methods, like max attack, but with significantly higher precision and a fraction of the computational cost.
Okay, let me pull these threads together for our third recap before we move to the final piece of the puzzle, because we've covered a massive amount of ground here.
It is a lot to take in.
Bullet 1, it crushes histone prediction, doing the whole genome in 2.5 hours. Bullet 2, it maps 4 types of functional elements using that cool peak calling algorithm. And bullet 3, it predicts gene expression and a massive 90 transcription factor sites all at once. It is essentially an all-in-one genomics Swiss Army knife.
And yet everything we just discussed is only the baseline of what makes this paper so groundbreaking.
Wait, there's more.
Oh, absolutely. Memorizing and predicting patterns on data you are familiar with is one thing. The true holy grail test of any deep learning model is how it handles the completely unknown generalization.
Right. And the constraints they placed on this model during training were extreme. Biosec-2thec was trained on exactly one single human cell line, K562.
Which is an immortalized leukemia cell line.
Right, that is the only type of cell the AI ever saw while learning.
Yet when the researchers took this AI and asked it to map completely different unseen human cell lines, like T cells from the immune system or GM12878 lymphoblastoid cells, it generalized beautifully.
Wow.
It accurately predicted their unique functional landscapes. But the researchers pushed it even further. They took this model, trained exclusively on human leukemia cells, and applied it across entirely different species.
Wait, hold on, I have to stop you there. A model trained entirely on one human cell line accurately predicted the complex genome of an entirely different species.
It did.
Because I read the paper, and it mapped mouse livers, horse livers, and even the ovaries of Drosophila, the common fruit fly. Honestly, that sounds like pure sci-fi. How is that even biologically possible when a human's DNA is so vastly different from a fly's?
Because the AI didn't just memorize the specific sequence of human DNA, it learned the universal underlying physics of how DNA sequence interacts with RNA polymerase traffic.
The grammar of the genome.
Exactly. It learned the grammar. The fundamental laws of transcription, how a promoter initiates a signal, how an insulator stops it, these are highly conserved.
Evolutionarily speaking.
Yes. Evolution figured out the mechanics of this operating system millions of years ago. The exact letters might change between a human and a fruit fly, but the syntax of how those letters form functional words remains virtually identical.
That is profound. But to ground this incredibly high-level biology into something tangible for human health, how does predicting the grammar of a genome actually help us fight disease?
Well, the paper provides a stunning real-world application using the IL-2RA locus. This specific region of the genome acts as a super enhancer in human CD4 positive T cells. Super enhancers are massive, dense clusters of regulatory elements that drive extraordinarily high levels of gene expression.
And they are critical for determining a cell's identity.
Right. And when they malfunction, they are heavily linked to severe diseases.
And CD4 positive T cells are the absolute command centers of our immune system.
Exactly. So when the researchers ran BioC2-seq to analyze these T cells, the AI predicted massive spikes in active regulatory signals right at this super enhancer region. Now, here is where it gets critical.
I'm listening.
They cross-referenced the AI's predicted map with the real-world GWA's catalog. That's a massive database of known genetic mutations in humans.
Okay.
They found that the exact functional regions the AI highlighted lined up perfectly with a dense cluster of real-world SNPs, single nucleotide polymorphisms.
And an SNP is essentially a single-letter typo in the millions of pages of DNA code.
Precisely. And the specific typos located in the region the AI flagged are clinically documented to be directly linked to 8 different human autoimmune diseases, including multiple sclerosis and type 1 diabetes.
So without being told anything about multiple sclerosis or diabetes, and without requiring a team of scientists to spend months running physical cheap P-seq assays on immune cells, the AI instantly pointed to the exact functional regions where these disease-causing typos occur.
Just by reading the natural traffic patterns of the DNA.
That is incredible.
If we connect this to the bigger picture, it acts as an incredibly accurate compass. It proves that Bio-Psych2-Seq can be used as a preliminary tool to connect genetic variants to actual physical biological mechanisms.
It totally democratizes research.
Completely. Imagine the impact. If a lab is studying a rare tissue type or an underfunded orphan disease, they no longer need millions of dollars for exhaustive physical mapping.
Right.
They can run one simple RO-seq assay to capture the live traffic, feed it into this computational shortcut, and hours later, they have a comprehensive, highly accurate map of the disease's regulatory landscape.
It is taking the vast, murky, overwhelming alphabet soup of biology and bringing it into sharp, readable focus. And it's doing it fast enough and cheaply enough to completely change how research is done.
It really is the start of a new era.
So I want to pose a question to you as you're listening right now. Think about the fact that an AI trained exclusively on a single human cancer cell can effortlessly decode the biological operating system of a fruit fly. What stands out to you about that? Does knowing that change how you view the connection between all living things?
It's a fascinating thread to pull on. If a deep learning model, which is ultimately just a complex sequence of mathematical weights and biases, can find the exact same shared patterns of transcription across billions of years of evolution, what does that tell us about the fundamental universal language of life itself?
Yeah.
Are the vast, incredibly complex regulatory networks that make a human so vastly different from a fly or a horse or a mouse ultimately just different dialects of the exact same code?
That is a wild thought to leave on. And suddenly, that infinite bowl of alphabet soup doesn't look quite so chaotic anymore. We finally have a lens wide enough to read the story it's telling.